Hello again, and welcome to another episode of Fitness Facts. I'm Dr. Jordan Taylor, the Undergraduate Exercise Science Program Director and Associate Teaching Professor at the University of Kansas. I have Jeremy Pearson back in the studio to discuss a recently published paper of his where he compared single set versus multiple set resistance training of the lower body and the different acute responses of several proteins within an intracellular signaling pathway that is known to promote muscle protein synthesis following training. Jeremy's a third-year PhD student here at the University of Kansas studying muscle stem cell activity in response to exercise. He is currently a trainee in the Gallagher Applied Physiology Lab and Rosa Moliner Microscopy and Analytical Imaging Research Resource Core Laboratory. Jeremy has published multiple peer-reviewed research articles examining how to build muscle and lose body fat simultaneously via exercise and nutritional strategies. In addition, he has been a competitive bodybuilder for over a decade. Welcome back to the show, Jeremy.
Thank you for having me back. It's a blast to be on here again.
A lot of fun. So let's dive right in and discuss the paper you wrote that was published recently in May 2023 in the Journal of Strength and Conditioning Research titled Similar Responses in the AKT Protein Kinase B Signaling Pathway Following Different Lower Body Exercise Volumes in Recreationally Active Men. Can you provide some background information on this paper and summarize the introduction of it?
Yeah, definitely. So kind of the background of this paper of why we wanted to do this is in the literature, from years and years ago, we see these conflicting results of how much volume do I need to increase muscle growth? And what we're seeing...
Let's define volume.
Volume, okay. Volume is
simple.
sets times reps. So how many sets am I doing? Like a set of 3 by 10. Am I doing 10 reps and then 3 sets of that, so 30 reps total? That's what we're talking about for volume.
Right.
And
how much work you're doing in the workout.
Yeah.
Basically.
Yep. So volume is kind of this heated debate, and even still right now, we don't know how much volume someone needs for inducing muscle growth or increasing muscle size. And some of the research has looked at doing just one set. So people that don't like going to the gym, can they go to the gym and just get one set done? Maybe they take it to failure. They just do one set and they leave.
One set of chest and one set of back.
Exactly.
One set for the bicep. One set for their quads. One
Is
set
that
for
enough?
hamstring.
Is that enough to induce muscle growth? Versus multiple sets. And from the literature, we're seeing conflicting results. Some show that, no, you need to do multiple sets. Some say, there's actually one study that we mentioned in here from Schoenfeld that one set was better than multiple sets, which is, that's
now, were those untrained or trained individuals? Do you remember?
Untrained.
Because again, in untrained, typically one set, it might be enough. But as you become more trained, you probably need more sets, more volume, more reps.
And I agree with that.
Because I can't imagine, and I'm just speaking out anecdotally here, if I cut my training back to only doing one set, I feel I would really be losing muscle mass and strength because my body is adapted to lifting 5 to 6 days a week, each muscle group twice a week. And
multiple sets.
Usually I could do 10 sets of chest in one workout. And then 72 hours later, I'm doing another 10 sets of chest, right? So I make 20 sets of chest in a week, 20 sets of back in a week, and splitting that volume across 2 sessions, So
definitely.
It's multiple.
Training status definitely.
But I've been training a long time, right? I'm 42. I've been training since I was 16. I wasn't doing that much volume on day one. That's for dang sure.
Yeah.
And when I was a personal trainer and working with clients and also a strength coach, like if I had a client come in on day one, we may only do one set or 2 sets for chest and for back and for the legs and for the arms, and that's it. That's where we're starting. So I think that's important when people, whether you're a student or you're someone in the general public, when you read studies, whether it's like this from Jeremy, Dr. Schoenfeld, anybody, Dr. Fry here, Dr. Herda, anybody at KU or anywhere, are they trained individuals or untrained? Because you can't extrapolate what you find in a trained group to untrained and vice versa, right?
I think that's a great saying. Just to reiterate that, if one study is showing that, this exercise program resulted in serious muscle mass gains, I cannot take that and say, these trained guys like us, let's do that program. It just doesn't work like that.
You look in the methods section, you find the subjects, read about these subjects. Are they male? Are they
Yeah.
female? Because that doesn't always, you can't
No.
always say what happens in male is going to happen in female
Yeah.
and vice versa. What's their age, right? Are they young or old? You know, you see different training responses in
Yeah.
young and old. Are they trained, untrained? So it's important to look at the subjects. And if you're comparing study A over here to study B, are the subjects the same or is it a whole different demographic?
Yeah. Mhm.
Right? Because that can affect the results and the conclusion of the study. But I'm stealing your thunder. Let's go
That's
back
okay.
into
It's okay.
kind of, again, why you wanted to do this study and what
Sure.
we're looking at here.
Yeah. So, like I said, volume is kind of this, debate right now of how much volume is enough. Can I do one set and get the same benefit that I'm doing from 3 sets? And that's what we wanted to know. We wanted to take a look if I can just do 1 set versus 3 sets. We kind of hypothesized at the beginning that 3 sets is going to be better for what we were looking at.
And now when we say what you're looking at, we
Yeah.
weren't looking at chronic
No.
a response over
Nope.
many, many weeks, months, or even years. We're looking at a single session, right? And
Yeah.
an acute response, and that's important. So you're looking at muscle protein synthesis, which is an acute response to either to a single training session.
We were looking at certain proteins that are involved with initiating protein synthesis. Yeah, to measure protein synthesis, it's a lot more expensive
Right.
and it's a lot harder. We don't have
Stu Phillips do that.
Yeah, I
right?
could just still do it. Yeah, honestly, yes. Yeah.
But yeah, I kind of jumped ahead, and we'll get into what Jeremy looked at as far as these different proteins and enzymes that are inside muscle cells that kind of get switched on, and when they're switched on, that causes these little organelles called ribosomes to start translating and bringing in amino acids, and that you form a protein out of it. And those proteins, some of them can get incorporated into muscle as a response to training. And then, like you said, the question is, well, can that whole mechanism happen with just 1 set, or do you need multiple sets?
That's what we wanted to know. We just wanted to know the acute response, if 1 set would be similar to 3 sets. From there, we can't extrapolate of what's going to happen chronically, but that's just muscle growth in general is multifactorial. It's not just, it's not one thing that's happening. And looking at these acute studies or looking at something acute, we can put more pieces to the puzzle to say, oh, this is involved with, chronic muscle growth, or this, here's one more piece that we can add that's showing,
And that's kind of getting at the big picture of why your research is important. And anybody that's doing research in this field, everybody knows you lift weights, you get bigger, you get stronger. Yeah, that's the duh.
But how does that happen?
But how does it happen? How do you get there? How can you optimize it? And you have to really know what's going on in the cell, as we'll get into this signaling pathway in a bit. And you have to understand how these different molecules and these enzymes and how they're all regulated and how they work together to create this muscle protein synthesis, producing these proteins, and how that's built into muscle. So there's a lot going on, but you have to understand that, and then how different training programs, hormones, nutrition, you get into all these other factors. There's just so many things to look at.
There's just so many, and that's why I'm doing acute research. I'm looking at, one of these different avenues that potentially, hopefully, leads to muscle growth.
Right. Well, let's talk next about what is an intracellular signaling pathway.
Sure.
Okay. Because that's wordy.
That's
intracellular
big. Yeah.
signaling pathway. If you had an undergrad student right here, and they raise their hand, they say, well, what is an intracellular signaling pathway? What does that do? What are you going to say?
Yeah, definitely. I would say that inside the cell, or even I'll take what we did. We have an exercise session. I exercise. I lift. I do a bicep curl or something. There's going to be this mechanical tension that's going to translate into a biochemical signal, and from that, there's these receptors on the muscle that are going to be activated when you have that, you do those bicep curls, you have that tension, and it's going to send these signals down this stream. You could call it a waterfall or a cascade of events. And if you're looking at the image there, looking at the green, that is where the receptor is, and it's passing, it's kind of talking, or it's like a crosstalk communication. It's from protein to protein to protein to protein. And eventually, when we say intracellular pathway, it's inside, so intra meaning within, it's inside the cell. So the proteins are going to be communicating within the cell, and it's going to be, an effect is going to happen. I could talk about how muscle growth is happening, but just for an intracellular pathway, there is an effect that's going to happen from all these proteins going to another, talking to another.
Protein activity, crosstalk. And a lot of it, basically, if you think about it with signaling pathways in a cell, like you mentioned, some sort of external stimulus. It could be mechanical tension from lifting a load. It could be various growth factors. So we have this, picture 2 that's pulled up here. We're looking at this AKT pathway that Jeremy looked at what was going on with these different molecules in this paper in response to either single set or multiple set training. And this pathway, it can really be, if you look at it, it can be activated by many things. Not only mechanical tension due to lifting loads, hormones like insulin, other growth factors, insulin-like growth factor can activate it. Certain anabolic hormones like testosterone can activate it. Amino acid levels in the cell, like for instance, leucine levels, can switch on a lot of these molecules inside a muscle cell. So what you're looking at here is you have a cell membrane, that's kind of the green circles there, and then you'll see that little arrow at the top with underneath insulin and growth factors. You could also put in, like I said, mechanical tension, other hormones like testosterone, IGF-1. Something is going to activate that receptor, right, of those different factors. And then once that receptor is activated, it gets phosphorylated. All these different little ovals that are colored, different colors, these are all kind of like dominoes or key players within the cell. And so when one domino is activated, it activates another domino. You think of dominoes falling over. And then at the very end of this domino cascade, you get protein translation that you see down there in that purple box. And that's where you're synthesizing protein. You're making proteins that could eventually get incorporated into muscle. Now, what Jeremy looked at, I kind of have in these red dashed boxes. So he looked at one of the dominoes or one of the molecules in this signaling cascade that can get switched on by resistance training or other things, growth factors, hormones. But now he just looked at, will resistance training switch this on? And what about single set versus multiple sets, right? So he looked at AKT, which is a kinase, an enzyme, and it has a little P on it in a circle, and that means it's been phosphorylated. It's been phosphorylated really in response to resistance training or growth factors or a hormone or something else. And once that molecule is phosphorylated and has that phosphate group tacked onto it, it's like switching on the light switch, or you can think of it in this case, it's going to make that domino fall over, and then it's going to activate things downstream of it. One of which would be mTOR, which is kind of in the middle. It's the central molecule there, mechanistic target of rapamycin. So this is kind of the master switch. That thing has to be switched on for muscle protein synthesis to occur.
Yes, that is one of the factors. That is one of the,
there's other pathways and crosstalk, but very simply, it's important for it to be switched on for muscle protein synthesis to occur. And AKT is upstream of that and can switch it on. So remember AKT, because this is something that Jeremy looked at. You didn't look at mTOR, though, because mTOR is hard to look at, that complex.
Yes. So in the paper, I even put that it is so difficult. Our lab has tried to quantify it via the methods that we used, and we just could not get it to work. So, that's why we switched. We went upstream, and then we went downstream.
Right. So you look at the dominoes above and the dominoes below. So now below mTOR, we have that S6K1. So maybe talk about what is S6K1's role. And you see it has a little phosphate tacked onto it. It's kind of switched on there.
Yeah.
So what is its role?
Sure. So I would just say both. So when mTOR gets activated, I'll start over. So with AKT, that was, that's right in the title of our paper, this AKT, protein kinase B, same thing. When that is activated, from this mechanical tension, you're from doing the single set or the multiple sets, AKT is going to swing down and talk to mTOR. It's going to activate that. When mTOR is activated, that's a good thing for muscle growth usually. And that's going to talk to P70S6K or the S6K1, and that is going to be a messenger that's going to be delivered to another protein that's right in the box. You can see it's the green 4-EBP1, and that one is going to initiate this protein translation. Or when I think of protein translation, I just think of what Dr. T said, protein synthesis.
Well, in 4-EBP1, because it's a binding protein, it binds that EIF4E, which stands for eukaryotic translation initiation factor, 4E.
Mm-hmm.
So and correct me if I'm wrong, but the way I remember it is 4-EBP1, it binds up that EIF4E, but when it's phosphorylated, the EIF4E is released, and then it can initiate protein translation.
Yes.
When 4-EBP1 is not phosphorylated, then EIF4E is bound up, and it can't go signal the ribosome to start cranking out proteins.
Yeah.
So that's why...
I wish there was a video, but yes, that's perfect.
The 4-EBP1 needs to get switched on and phosphorylated, and mTOR can switch it on. But again, mTOR has to be switched on by AKT. S6K1 is important for translating proteins or muscle protein synthesis as well. But yeah, that 4-EBP1 has to have that little blue phosphate group tacked onto it so that the EIF4E can move to the ribosome and then allow that muscle protein synthesis, that translation to occur.
Yeah, and mTOR, so those two arrows from mTOR, the mTOR can signal a lot of different things as well, and S6K1 can also signal a lot of things as well. So there's a lot of arrows coming out.
Yeah, think of mTOR standing there with like a megaphone yelling out all kinds of stuff, and a lot of different downstream people or molecules can hear it. But,
just a waterfall effect of all this.
I think it's, again, important to just remember this is a cascade of dominoes. One gets switched on. A lot of times it's switched on by phosphorylation, a phosphate group getting added. That kind of turns it on, it'll cause the domino to fall. Like you said, it talks or knocks on the door of another molecule. And again, a lot of these molecules are proteins, and some of these proteins are enzymes that catalyze reactions. And then mTOR switched on, then you have those downstream molecules you looked at, which was S6K1 and 4-EBP1. So again, just to summarize, you looked at AKT, upstream of mTOR. You looked at S6K1 and 4-EBP1.
4-EBP1, yep.
So upstream and downstream of mTOR. Are these being switched on? And if so, will a single set do it? What about 3 sets? Do you get more activity in this pathway with 3 sets compared to 1? All right, these are some questions you looked at.
That's a beautiful way to put it. Yes, absolutely.
So looking at the activity of this pathway that leads to muscle protein synthesis, and hopefully, if you're getting spikes in muscle protein synthesis each day you lift, and you do that over weeks, months, and years, hopefully that you get muscle growth, right? That's what you're after. So I think we covered it there. Right?
I think
that's a
good.
complicated pathway, but I think we
don't get hung up on the pathway.
Enough,
just
right?
Don't get hung up. Yeah.
So let's talk about. Well, we kind of talked about it. Just state very simply the purpose and hypothesis of your study.
Yeah. So the purpose of our study, our study, was to look at the differences in volume, one set of leg press versus multiple sets, 3 sets of leg press, and to see how it would affect a signaling cascade or a signaling pathway that is heavily involved in chronic muscle hypertrophy.
Which we just talked about.
Yes.
The AKT pathway. Yep.
What did I think was going to happen? What did I hypothesize? We thought initially that 3 sets would have better results, have more protein quantification for those three proteins that we looked at. But overall, just once we that was initially when we started writing it and more data came out, we didn't think there would be any differences.
Right.
So that's where I'll leave it at.
Okay. So let's talk about the methods and the procedures used for this study, right? So maybe first we'll talk about the experimental approach. Let's talk about the subjects, right? Always start with the subjects. So, again, we're going to compare, like you said, 1 set of leg press versus 3 sets of leg press. I'll let you kind of take it from there.
Yeah, definitely. Yeah, so when we're talking about the subjects, we had 8 that were going to do the single set, 8 that were going to do the multiple set. All of these, they were recreationally trained guys, so they were, Honestly, I think in the methods I put that they trained at least three
to
or
four
times,
sessions per
sessions a week.
Yeah.
So yeah, they were,
They were not overweight, so they were normal weight.
No, yeah,
Okay.
yeah.
Didn't have any metabolic disease. Yeah, no conditions. Okay.
I would say they were moderately trained college students.
Right, and generally healthy. Yeah.
And healthy, yeah, moderately trained healthy college students. Yep.
Okay.
Okay, so we got their 1 repetition maximum on this leg press device, and
That's
the leg
your baseline
press.
testing. Get their 1 rep max. You got to know how strong they are before you start the study, right?
Mm-hmm. And this leg press, some of you might not be familiar with how or what device we use. We use a Pneumatech. It's basically an air pressure device. You guys might have heard of like Kaiser instruments, but it's an air pressure automated leg press. Instead of a plate loaded or like a pin loaded leg press, it's using air pressure as the resistance.
Right.
And it's applying resistance during the positive movement, the pushing, and the negative or the concentric or the eccentric, if you're familiar with that.
So you got their 1 repetition max, which is important. You got to know everyone's baseline strength, and then talk about the different resistance exercise protocols because you divided the subjects into groups. So maybe talk about the groups, and then what percentage of 1 RM were they going to work at? Kind of how the study design there with the exercise protocols. Yeah,
definitely. So like I said, there's 2 groups: the single set, the multiple set group. The single set, pretty easy right off the bat. They're training at 80% of that 1 rep max we found on the leg press. And they're just going to go to failure. It is 1 set, all out, gun to the head.
Right.
How many can
Give
you
me
do?
1 more.
Yeah,
right?
exactly. Yeah, we're yelling at them. Yeah, absolutely.
Right.
The other group is 80% of the 1 rep max. The multiple set, and they're doing the first set to 10 repetitions, done. The second set, 10 repetitions, done, and then the third set to failure. We put the gun to the head and say, go, come on, let's do it. Yeah.
Right. So there you see your single set versus multiple set groups.
Yep.
All right. Okay. Now, after they've went through this acute, resistance training protocol, these 2 groups, you needed to collect some tissue, right?
We needed, what are we going to look at? Exactly.
Right, so obviously they were doing the leg press, which works the quads, so you got to get a sample of quadriceps muscle, right? So let's take a look at this video where we're going to get a sample of the quadriceps muscle, after the resistance exercise. Now you weren't a subject in this, but you have had.
I have had. This is me.
A sample taken before, so
I don't really like looking at this.
But this is in the Gallagher lab. We got Dr. Gallagher doing the biopsy here, but you were a subject for this. So what are you thinking here?
Well, I'm very scared right now. I don't even like getting my finger pricked, but I had to do it right now. So Dr. Gallagher kind of injected a bunch of lidocaine, so I'm not feeling anything. And honestly, overall, he
was kind of poking to see if you could feel it, right?
Exactly.
See if you were numb.
Yeah, and I didn't feel a thing.
Right.
I didn't feel a thing. If you can't hear the audio, I even say I can't feel a thing. And honestly, when he made that incision, I didn't even feel that.
Yeah.
I felt nothing.
You were numb with the lidocaine, little inch or so, half-inch incision.
2 centimeters.
Yeah. Okay.
Yeah, not even an inch. Yeah. Yep. But yeah, so he just made the incision. And then
here's the
biopsy
needle.
There it is.
It's thick, right?
And I'm probably sweating and about to cry, but everything's fine. It worked out. But and it's really not that bad. It just looks scary.
And then you're using suction, so he's going to do a punch.
So he
suction.
punches. He's going to go up, and I'm sucking, or my lab assistant is sucking, and then it's done. That's it. So in that, do you mind if I have that for a second? So
talk about the biopsy needle.
Yeah. So with this needle, it's 2 compartments. So there's the kind of the main part that we're holding, and then this outside portion that is kind of goes into it. And right here, you'll see that there's this open hole that is when we go in. Dr. Gallagher, my mentor, is going to open it. And then suction is going to be applied. The muscle is going to go into this, this cannula, this hole, I guess, right? And then Dr. Gallagher is going to go snip. And again, I really don't feel that just because of the lidocaine. I feel kind of like just a bunch of pressure. That's Yeah, but so I was scared for nothing.
Well,
So
now of
don't-
course the lidocaine doesn't make its way down to the muscle. It's the dermis and subcutaneous tissue. But
he does go into the muscle.
Does he?
Yeah,
Does he
yeah,
go?
Yeah. So
All
what
right.
he does, actually, that's a good point. So if this is my leg or whatever, he's going to go a small injection to make a bubble in my skin and then the dermis, and then once he goes that, he's going to go straight into the muscle. And
Okay,
that one does
okay,
not feel
all
good.
right.
Yeah, I don't like that
yeah,
one.
okay, yeah,
Yeah.
you're gonna get a good burn. Yeah.
That one
Good
feels
deep-
like a bee sting
good deep
inside.
burn.
There's like a like a worm in my leg.
Yeah.
That's kind of mean. Yeah.
Okay.
Yeah.
So
But-
yeah, so I guess so. Then now, if we pull up this next picture here, you can see in the petri dish. Talk about what we have there.
So like I said, when it's in that hole and we snip it just like that, he'll grab the needle out just as you saw in the video. He'll bring it over to his desk and he'll use this other little. It's a kind of like a metal needle that goes into this hole right here, and it's going to push the muscle outside of this hole. And then we just grab it with the tweezers, and there you have it. That is my muscle tissue.
Some tasty little steaks.
Looks pretty
there,
good, right? Yeah. Yep. Everyone, I've had it before. You know, my family's like, "Oh, I didn't know it was actually red." Yeah, muscle. Yeah, it's red. Yeah,
Right.
it does look a little. I don't know if you have the images later of the muscle fibers, but it does. It's a little clearer.
Let's look at because you've got to process the tissue, right? I mean,
Yeah.
So let's look at the next picture
Yeah, there you go.
under just a light microscope. I
Uh-huh.
mean, you've got individual fibers there.
Yeah, so if you guys can see, look at, this is a huge bundle. There's hundreds and hundreds of fibers there, but you can see one that kind of looks like a hair string off. That is one muscle fiber. So if you guys see that on the bottom right, yeah, perfect. I love it. Yep, that is one muscle fiber.
Yeah, that's awesome. So let's talk about you've got this tissue now. You collected tissue via this method, this punch biopsy for all the subjects. Now, when did you do these biopsies, right? You did it at baseline.
I needed to do it at
You do it
baseline. I need to
baseline
see how
before
it
they do the
changes.
exercise,
Uh-huh.
before they do the resistance training. Then when do you collect the other samples?
We did, if I remember right, it was 15 minutes and then 180 minutes.
Yep.
Post. So
Post-exercise.
Post-exercise. Yep. So we wanted to look at specifically for those time points. We know that gene expression or protein translation or just kind of like protein activity is very prominent during those time points. We don't want to wait a whole day. We don't want to wait, right after. We want to wait a little bit just so, just in the literature, Dr. Gallagher has actually published papers on this, that gene expression, looking at protein stuff, that's the key times is 180 minutes post, 3 hours post, or 15 minutes post, 20 minutes post.
Right, and remember, if you remember back to basic biology, it's like gene expression. That's where a piece of DNA is copied into this little mRNA molecule. The mRNA leaves the nucleus of the cell, goes out to a ribosome, and then the ribosome translates that and creates a protein. So the gene expression is really just the copying of a gene, and that gene being turned on, and then the mRNA being translated into a protein. And there's different time points that you want to try to hone in on, because you can miss the time points sometimes. If you don't use the right time points, you may miss the effect you were looking to see because you didn't have the right time point, and there could have been a response, but you missed the time point because you waited 8-9 hours instead of 15 or 180 minutes post-exercise to look to see what's going on with the genes and the proteins in the cell. And a lot of those proteins we mentioned that you looked at that were in this AKT pathway. You looked at AKT, you looked at the
P70S6K,
the S6K1 and the 4EBP1, that binding protein. So those are the different proteins. He was trying to look at their activity, their expression level actually, with these samples, right? And so then maybe talk about, now you've got the tissue like we saw that you collected, and you've got to homogenize that tissue, you've got to kind of grind it up, and then what are you going to do when you've processed that muscle tissue from the subjects that are in these two different groups? What's the next step?
Yeah, sure. So there's two different things that we did. And I kind of mentioned it before. So there's gene expression that we want to look at. So how, like, kind of that transcription translation of for gene expression, at least we want to see how specifically how much of this gene is expressed, how much of this gene is upregulated or downregulated.
And when we say this gene, again, it's the AKT, the S6K1, or the 4EBP1. We're looking at, are those genes switched on, like in the nucleus?
Yeah, is that gene being produced more?
Right,
yeah, is
exactly.
there more production of that? Exactly, yeah. That's the way I look at it. It's kind of like a factory of, like, pushing that stuff
Think
out.
of it as a copy machine. It's just cranking out copies.
Right, yeah,
because that's literally what mRNA is a copy of that gene.
Yeah.
And is the resistance training switching on that copy machine to crank out mRNA molecules.
Yep. And so that's gene expression. Yeah.
Right, that's gene expression. So then, and that's occurring in the nucleus of the muscle fiber cells in the nuclei. So then what did you look at? What technology is used to analyze gene expression? You don't have to go into depth about it.
Yeah.
But, I ran a ton of these at KU Med Center, but maybe what I was looking at brain tissue, but
nice. Okay, yeah.
What is used for analyzing these copies, this gene expression?
So you might have heard it. It's called PCR, or real-time quantitative polymerase chain reaction. So basically, what we do is we take that muscle homogenate and we take it into a test tube and we put a couple of different things in it. We put ultra pure water. We'll take, the way I explain this to my undergrads, if that's okay.
Oh yeah, explain it that way.
Okay, great. So there's a funny way to explain it, but if on this table, I have all these different genes in my muscle tissue, and I want to look at a certain gene. I want to look at 4EBP1, I want to look at mTOR, I want to look at AKT. I like thinking of those genes as they're a bunch of different Legos. And if I want to look at a certain one, I need a special Lego that's going to connect to it that I put on. Mm-hmm. So we call that a primer probe set. The primer probe is going to latch on to that specific gene sequence, and then it's basically isolated. I can take that and I can look at how much of that gene was expressed, or how much of that gene was in that tissue.
Expressed as a result of the either single set or multiple set training.
Yeah, I love it.
Tie it back in,
Yeah, for sure. Yeah, so exactly.
Is there going to be a difference in the amount of that copies made of that gene in single set versus multiple set groups?
Right. So looking at that, I take that primer probe set, those Legos, I put it in that Eppendorf tube, and then I'm going to spin it down and I put it into this, this machine that's basically going to spin really fast, and it's going to look at the fluorescent integrity. And I don't even probably need to go into that, but it basically just analyzes how much gene activity or how much gene expression is in for that specific gene, that specific Lego that we attach to.
Right, because the protein is the final product. A gene has to get expressed and switched on for these different proteins to even be produced, right? And then next, which leads into talking about protein, you did Western blots to analyze the expression, and I guess you could say it's a semi-quantitative way.
It's a quantitative way for how much protein was actually translated.
Of translating, how much of this AKT was produced, how much 4EBP1, how much S6K1, these different protein molecules in that signaling pathway we discussed earlier. You're trying to quantify that through Western blotting. So maybe you can talk about Western blots.
Yeah, and even before I do that, what you said about how gene expression is happening inside the nucleus, that's a great segue. So the gene expression is happening inside the nucleus. It leaves the nucleus and it's now into the cytoplasm. It's not necessarily being translated. It's not necessarily creating a protein. We hope it will, but that's how we're checking it. That's how we're going to look at it. We're going to look at, so it's in the cytoplasm. I'm going to look at how much protein is being produced from using this term we say Western immunoblotting, or Western blots, immunoblotting.
Right.
Basically, what I do is I grind up the tissue. I take the samples and I'm going to zap it with electricity. The protein is going to run on this gel that I made. It's going to kind of run on this, I think of Jell-O. It's going to run through this Jell-O, and the reason it does that is all the different proteins that are in muscle are different sizes. They have a different molecular weight.
Yep.
And from that, when I zap it, the big proteins are not going to run that far. They're really heavy, right? So mTOR is one that's really, really big, really heavy. The small ones, like 4-EBP1,
Lighter proteins.
lighter protein, it's going to run. It's going to run real fast through that gel.
So the electricity, the current, basically helps separate these different proteins
Uh-huh.
he's wanting to look at in this signaling pathway by weight. They're separated by weight.
Yeah. And
Yeah.
okay, so now that I have all these proteins separated in this gel, I want to look at the specific gels. It's kind of like isolating out. It's kind of like that Legos thing that I said. I need to attach something to it. So where it comes from immunoblotting is after I get that gel, I'm going to put a primary antibody onto it. And actually before that, I'm going to block it. So I'm going to block all of these proteins. I'm going to, I'm going to put all the, yeah, I'm going to isolate. Yeah, I'm going to isolate all of those. And then after it's kind of like, I think of putting like a blanket over a table, then I can set the table with my primary antibody. My primary antibody is what's going to bind to that specific target. So if I want to look at the 4-EBP1 or AKT, that's what I'm using is a antibody, a primary antibody that's specific.
It's going to bind to that.
Exactly.
Yeah.
It's specific for AKT. It's not specific for AMPK or TSC1. I don't care
Right.
about any of those. I want AKT.
Right.
So I do that. I put primary antibody, and then to see it, to visualize, I need to put a secondary antibody onto it. I think of like a Christmas tree that goes onto it. So if I have my AKT here, I put on my primary antibody, but I still can't visualize it. So then, after that, I'm going to add a secondary antibody, kind of like a flashlight that allows me to
lights
see
it
it.
up, illuminates it.
Yep.
And then that way, that's what allows you to measure the protein expression.
Yeah.
Right?
And
Because you have to be able to see it to quantify
Mhm.
how much was there in that muscle tissue.
Yep.
Yeah.
And that's how I
in
quantify
response
it.
to the single set versus multiple set.
Yeah.
And again, think of it this way. It's like, so you do exercise, right? That's going to cause something to happen, maybe, potentially, in the nucleus of, and muscle is multinucleated, in the nuclei of that trained muscle. And that switches on specific genes, and those genes make proteins. And it's the proteins that are actually the players that really do the work in the cell. Some of these are enzymes, some of them catalyze reactions. Some proteins that are made actually become contractile filaments in muscle. But I always think of like gene expression, it's kind of like a coach on the sideline telling the team to do something.
Yeah.
The players on the field are actually the proteins that are going out and doing the work. And that's kind of
Definitely,
how
yeah.
it is in your cell.
Yeah, that's a good way to put it. Yeah.
So,
Absolutely. Yeah.
well, I think this is great. I mean, now we'll maybe talk about, let's move into the results. And you can kind of talk
Okay.
about the stats as we go into the results.
Sure.
So maybe we'll pull up, this is picture 5 here, table 1. So this is pretty simple to explain. Just explain this table. You've got your
Yeah.
groups, just so everybody can see. Maybe go through each column there.
Yeah, definitely. So looking at it, it was just kind of looking at the volume and the load of the differences between the groups. So SS is the single set, just the group that did one set to failure. MS, multiple set group, they did the 2 sets to 10, third set to failure. And then I just combined them just to show the total. But yeah, okay, so looking at the group, and then on the next column we have 1 rep max. So we have their, when I said when they first came in, I got their 1 rep max from that leg press. Not much different, right?
Right.
There's not, so they were pretty similar.
Similar starting baseline.
It wasn't like you doing a leg press versus someone that doesn't work out, right? So
right.
they were
Because
pretty
they were all
similar.
recreationally trained, about moderately trained individuals.
Yep.
So, yep.
Yep. And then looking at, so the next column is the actual session. So the single set group did 26 on average reps at 80% 1 rep max, which is actually really high.
Yeah, because normally you would think if you're lifting a weight that's 80% of what you can lift one time, you're probably going to fail between maybe around 8 to 10 reps.
Yeah, yep, 10 reps, yeah.
But that's probably because it was a pneumatic device
Exactly,
and
and
not
that's-
a typical like plate-loaded leg press. It's because the device was a little different.
It doesn't have momentum, which is the weird part. So it's kind of like you press, and then it comes back, and then it just it's different.
Right.
It's different, and that's one of the limitations that we talked about in the study. That using
Right.
this Pneumatech device, which we probably should have used a real leg press, but it was a brand new piece of equipment. We wanted to try using it to see what it was like.
Right.
Dr. Fry bought it, but a lot of different rehab places, a lot of different actually facilities. The University of Tampa had a ton of Kaiser Pneumatech.
Right.
So it's actually rehab centers I think are coming out with a lot more.
Yeah.
Performance centers are using those a lot more. But
It
anyways,
could be safer without having that momentum and things.
Yeah,
to use in a
potentially,
rehab
yeah,
setting.
potentially.
I mean, thinking. I don't know if there's any studies on that, but yeah, I mean, you would think it could be.
One of the big reasons was because these guys were pretty well trained. It saved us a lot of time having to load the plates
That
and
would
stuff
make
like
sense,
that.
yeah,
You know, I
yeah.
literally just went
Yeah.
like that. That was it. Like I just took the switch and I'm like,
Crank
okay,
the knob.
add 300 pounds.
Right.
Done.
80%. Get it there.
Yeah. Like that was nice. Okay, so looking back at it, we can tell that the single set group did 26 repetitions. The multiple set group did way more than that. They did 47 reps, which makes sense, right? So they did the last set to failure and then the 10 reps. So you had 20 to it. So it was pretty similar.
Right.
So,
Okay.
and then there was a significant difference between those, which we'd ran a t-test and that makes sense.
Right.
You know, there was a significant difference between the two groups for the reps performed.
Yeah, multiple sets, you're going to get more reps.
Yeah, and honestly, I don't even need to talk about the last. It's just the
Right.
total volume load. It was higher with the multiple set
Yeah,
group.
weight times the number of reps.
Exactly, yeah.
So, yeah, it makes sense. Multiple sets going to have more.
Yep.
All right. Well, then let's go on to picture 6. So this is figure 2.
Yeah, so this is AKT? Yeah.
Yeah, so here, talk about what we're looking at. So again, remember everybody that AKT is upstream of mTOR, that switch that needs to be on it's part of the process involved in muscle protein synthesis. So this is the molecule in this intracellular signaling pathway that we talked about earlier that was in one of those dashed rectangles upstream of mTOR that you can talk about it, AKT here.
Sure. Yeah, so whenever I teach my students or even just, my research assistants this, I like saying that when there is a figure, there is a graph, I always like looking at the X and Y axes first. So looking at the X axes, that's our time points. So I did the pre-biopsy, I did the 15-minute biopsy, and then the 3-hour biopsy. So those are the time points of when we took the biopsy.
When you got your muscle samples.
Yeah,
right? Baseline, 15 minutes post-exercise, 180 minutes post-exercise.
And then the Y-axis is the value of that protein quantified. Okay, hopefully that makes sense. So we're looking at phosphorylated AKT. That is basically the total amount of protein that was phosphorylated.
Right, that had one of those little blue balls with a P in it that we showed on that graphic earlier.
Yep.
So how much had that little blue ball, that little phosphate group tacked onto this AKT protein?
Right, and if you look on the bar graphs, we have the white and then the black. The white is the single set group, right? Just that did one set to failure. The black is the multiple set group, the 3-set group. And from there, we don't see, there's no asterisks, there's no like stars or anything to denote that there was significant differences. Even though it might look like it, the values just do not reach statistical significance. So overall, we're not seeing a difference. And actually what is better to look at or more appealing to I'm a very visual person, not a numbers guy, but if you're looking above, those are the Western blot images. So we call them the blots.
Right, that have been labeled with the antibodies tagged. They're kind of lit up there, those little lines that you can see.
Exactly, yeah. And just how I said that before, I have my sample, I have the muscle tissue, I have the probe that I want, the AKTs right here. How do I label it? I put the primary antibody on, I put the secondary antibody on, and then from there, I can image it. And that's the image right there. So you're looking at these little wells, or they're little like blot images. We just call them blots.
Yeah, and you're kind of looking at intensity.
That's a good point. Yes, yeah. So we're looking at how intense. I wish I had one to compare it to, but basically, right there you can tell that there is protein there. There is, a decent amount of protein there. If there wasn't, you would see kind of just a super faded black background. But from this, I'm seeing pretty much the same intensity all over the board.
Yeah,
so,
and generally it all looks similar, regardless of the time point. 15 minutes, 180 minutes. It doesn't matter if it's single set or multiple set. It all looks, the intensity of the light there looks very similar.
Right.
So, and again, why are we looking at phosphorylated AKT? It's because it's the phosphorylated AKT that is switched on that signals downstream to mTOR which switches on mTOR, which then signals down to these other proteins that we're getting ready to
That's
look
the
at.
active one. Yeah.
So we can go to the next figure, and that would be picture 7. So now we're looking at phosphorylated S6K. So maybe talk about this one. Now this one is downstream of mTOR.
Yeah, downstream of mTOR, and it can be, it can signal all different other proteins as well, but S6K is heavily involved from mTOR. It's allowing basically just this messenger system to crosstalk, to communicate with other proteins downstream that will initiate this protein translation or protein synthesis.
Right. When we say protein translation, that's literally reading a copy of mRNA, a gene that's been expressed in a ribosome, linking amino acids together to make a protein.
Yep,
right?
To create different amino
like
acid
translating
streams.
what you're reading. I mean, that's literally what it's doing at the ribosome. So yeah.
Yeah, but again, so with this, same thing, same setup. So the x-axis is the time points, y-axis is the quantity or the values of this protein that we blotted and looked at. And really, again, it looks like there's significant differences from the bar graphs. But when you look, honestly, so you might see it different from the groups. But looking at the time points, there's really not much of a change. And from the blots, too, you're not seeing much of a change.
Yeah. A lot of times what people have to keep in mind is like when you see bars like that, and you're just like, well, wait, the single set group is higher than the multiple set. But there's a difference between it being statistically significant and plus the error bars on some of them, they're kind of big. So that's why you probably didn't reach statistical significance there.
Well, and just looking at the time points. Looking at the white, like there's not much movement from pre to 15 to 180.
There's not.
Right.
That's true.
Yeah. So overall, not much difference. And so going back to this whole thing that we're doing, there was no difference between a single set versus multiple sets for this protein translation or this protein quantification.
For this one. And then, we'll go to the picture 8. So this would be figure 4. Now here, you'll notice we've got our bars, the same x-axis, the same time points, the y-axis. Now we're looking at a different protein here. So you want to talk about this.
Yeah.
This is downstream mTOR.
Yep. So this is downstream of mTOR, the 4-EBP1. That's the one that, we phosphorylate, and it's going to break off from the 4-EIF, and it's going to kind of go into the cell and initiate.
It allows translation to occur. Allows EIF4E to do its job, which is promoting that translation. But if it's not phosphorylated,
it just stays.
Then it's bound up to that translation initiation factor.
Yep. Yeah. So we need that phosphorylate. I know it's a scary word, but it is very important.
Yeah. If you're ever a student, grad student, if you're ever taking any molecular biology class and you're just wondering what the answer is inside of a cell in a signaling pathway, just put phosphorylation down. It's probably
there you go.
Things are either being phosphorylated or dephosphorylated, and that's what kind of is switching on or off things in the cell. So talk about what we have going on here with this 4-EBP1.
Yeah, so what we're seeing is a significant difference from pre, but no significant differences. So it's really just showing that if you did any sort of exercise, the single set or the multiple set, that you're going to increase the translation or increase the amount of protein of 4-EBP1. But as you can see between the two groups, there's no differences.
Right. So at 15 minutes post-exercise, whether you did a single set or multiple sets, you see an increase. You also see it at 180 minutes. And it's regardless of which type of protocol you did, the single set or the multiple set.
Yep, exactly. Just anything, just increase this protein.
So the 4-EBP1 is being switched on, which then you'd think that's going to allow muscle or translation of proteins to occur in the muscle.
Yeah, so with the next image, the next table, this was our gene expression data, and we looked at upstream. So if we can go back to the first image, is that picture 2?
Go back to picture 2. Yeah.
I think IGF-1 is way up.
Yep, so maybe talk about IGF-1 and the receptor there that's in the cell membrane. The cell membrane is those kind of green circles there. So maybe talk about that.
Sure. Yeah. So on top there, if you can see, the IGF-1 is way upstream from AKT, from mTOR, from all of the other proteins that I looked at.
It's like the starting point.
Yeah, it is.
Pathway.
Yeah. Yeah. And IGF-1 is a protein, and it also has a receptor. And once the protein is, activated and it binds to the receptor will send these messages out.
And relay it to this whole cascade,
exactly.
This whole pathway. That's the intracellular pathway. It has to get activated by that receptor being activated by either IGF-1, resistance training, a growth factor, a hormone like testosterone or insulin. Yeah.
Yep.
Something has to activate the receptor for all those little ovals and everything inside the cell to be active that leads to protein translation.
And it's so far upstream, that's why we wanted to look at the gene expression of it.
Yeah. So now I guess we could go back to that table, picture 9. So, and I'll let you talk about what you looked at here.
Yeah. Okay. So again, the groups, we're talking single set and multiple set. The IGF, so the next column over, that is, we're looking at, I remember this. So when we analyzed the gene expression data, it was only pre and only 180 minutes post-exercise. The reason we did that was just because gene expression peaks at 180 minutes for most genes. That's why we only did 2 time points. The 15 minutes post, it wouldn't have even really shown
right.
much.
You wouldn't have had enough time yet for, for that gene expression to be activated. Yeah.
So looking at, those are just the raw values of I think the cycle thresholds, just the values of that gene for the
I remember
IGF.
calculating this. We don't have to talk about how it's calculated.
We don't have to
the
talk
delta
about that,
CTs.
There it is. It's right down there.
About it.
Yeah. But anyways, so there was a significant difference for IGF-1 receptor. There was no differences looking at the 8.89 and 4.74 for the multiple set versus single set.
So IGF-1 hormone, insulin-like growth factor hormone, there was no differences,
protein,
right?
But the receptor.
The receptor it binds to, there was differences between the single set and multiple set.
Yeah, and oddly enough...
Or I should say the gene that produces the receptor.
Yes, right. Oddly enough, we found that the single set was significant over the multiple set.
That's interesting.
Do I have a reason for that? I don't.
Yeah,
I don't know why we found that.
Well, you know what's interesting about IGF is there's different forms. So typically, I'm just giving a gross overview for everybody here, but the pituitary gland, way up in the brain, produces growth hormone. Growth hormone gets released in the blood, goes to the liver, tells the liver, knocks on the door and says, hey, make IGF-1. And then IGF-1 has lots of effects on pretty much every cell in the body. I mean, it doesn't matter if it's cartilage or bone or muscle. IGF-1 can promote growth. It has a lot of different effects on cells. But for it to have these effects, it has to bind to the receptor. And that's what else you looked at here is the IGF-1R receptor. But what's interesting is, there's different variants of IGF-1. Like you have produced locally in muscle. This might be something to look at down the road is mechanogrowth factor.
Yeah, definitely.
Mechanogrowth factor doesn't even get released into the blood. It works in an autocrine or paracrine manner. So it means it can be produced in a muscle cell, bind to the same receptor on that cell, and then initiate those signal transduction cascades within that same cell. Or it can be produced in a cell and knock on the next door neighbor, paracrine.
Yep.
Bind to the IGF-1 receptor next door and maybe activate, again, the signaling pathway in the neighboring muscle cell.
Yeah.
But it's not even released in the blood, I mean, it works in that autocrine paracrine manner, but that's mechanogrowth factor.
Right. Yeah, that would, that's definitely something we can look at. It's worrisome, or it's potentially, we don't know if we have certain primer probe sets for that it's good with muscle. Like it's kind of like a methods thing. But it's something to look at. It's definitely something to look at.
Yeah. So I think we'll move on to question 6. So can you discuss the limitations of this study and what conclusions were drawn from this research?
Sure. So right off the bat, I mean, it's small sample size. That's with, a lot of studies like this. It costs a lot of money to do these things. It's people don't want to get biopsies for free. So, 8 subjects, it's semi-low. It's pretty low. So, 8 and 8, that would have been nice to have more subjects. And I mentioned this before, the Pneumatech, the leg press machine, that was definitely a limitation. I mean, that's just we shouldn't have been getting 27 reps for an 80% one rep max. You know, that's just way over the board. But anyway, so those are kind of the main limitations. You know, we could have
another
looked at,
limitation. I'll think of it because I was rereading the discussion, but
Yeah.
you know, you looked at IGF-1 gene expression at 180 minutes and didn't see a difference between groups. You did with the receptor, but not with IGF-1. But it may have been a time point thing. You looked at it at 180 minutes.
Yep.
If you had looked at it, like it says here, you know, maybe if you had looked at it, there's some research showing that IGF-1 gene expression response does not occur until between 3 and 9 hours post-exercise. So you looked at 3 hours. Maybe there wasn't enough there.
Yeah.
But then if you looked at, say, 6 or 8 or 9 hours post-exercise, then you saw it switched on.
Time points of the biopsy, yeah.
And, you know, there's limitations there. That doesn't mean it's not a good study. It just means, you know, there's other time points to look at in future studies.
Definitely.
Right? So...
and
then you were talking about limitations with now the leg press, right? Is that where you're going?
Yeah.
So we kind of talked about that earlier, how
they shouldn't have been doing 27 reps for 80% one rep.
Right.
That shouldn't have happened. But, you know, overall, you know, more work was generated. They did more volume than we would have imagined. You know, is IGF, or is not IGF, but 4BP1 and P70S6K, AKT, is that stimulated by load or volume? I don't know the answer to that. So, you know.
You have to have multiple groups and look at different intensities and volumes down the road. That could get really, you know.
That sounds like a nightmare.
But be interesting stuff to look at.
Oh, yeah,
for sure.
For sure.
And then I think, you know, any other, kind of talked about the limitations. I guess we get into the conclusions and then the practical applications. So we have this this last picture here, picture 10 up, this figure. You know, folks can read that, but what are the practical applications? What can trainers, coaches, practitioners, anybody interested in this area, what can they take away from this study?
Yeah, definitely. I would just say, for anyone listening, if you don't really like working out, I think, and you don't work out a lot at all, if you're, you know,
novice.
If you're a novice,
right.
Definitely.
Untrained novice.
If you don't like working out, I think you can get away. You can actually make significant muscular adaptations from doing one set to failure per week. Is it optimal? Probably not. You'll probably have to progress that later on, but you know, from starting and doing, you know, maybe you know, a couple months of doing one set to failure per body part, I think you can make a semi-significant you know, I think you could achieve muscle growth, and not from this paper, but there's other studies showing that one set was enough to induce muscle growth. So
our data suggests one set to muscular failure versus 3 sets of lower body resistance exercise can elicit similar acute anabolic responses in recreationally active men. Remember, that's so we don't know about women, we know about men, and they were young. Specifically, performing one single set to failure of lower body resistance training may be able to achieve similar acute hypertrophic signaling outcomes based on those proteins you saw switched on or expression changes. Versus compared with multiple sets, potentially recreationally active individuals may be able to evoke a significant, I guess the one you saw switch on was primarily 40BP1, right? That you saw the significant differences in. Potentially, these individuals may be able to evoke a significant acute response that hopefully translates to chronic muscle hypertrophy by strength training at low volumes compared with having to do longer multi-set volume workouts. As long as you're performing that one set to failure. I think that's a key thing. That one set, you can't just do one set and be like, oh, I'm just going to do 5 and stop. No, it's got to be, you got to take it all the way. Like that you cannot do anymore. Because some people don't know what failure is. It's like, no, you have to do this until you literally cannot produce the force necessary to overcome the load. That's muscular failure.
Yeah, to where you cannot get another rep. You're doing bicep curls and you cannot go from the bottom you cannot go up.
And a lot of people don't know where failure is. You know, people talk about reps in reserve. I've got 1 or 2 left. Well, how do you know how many you have left if you've never actually went to failure? You don't. So, you need to go all the way to failure. Now, remember, I think it's important. This is an acute. We're looking at an acute response of these proteins in this pathway that can lead to protein translation in muscle. But to really know if it leads to muscle growth over time, you would need to look more at chronic, long-term training.
Definitely.
But it's a start. It's looking at a pathway. It's looking at mechanisms here.
A lot of studies are doing that, where they'll do one acute bout of exercise and they'll look at the gene expression, the protein, like immunoblotting of that, and then they'll continue that. They'll train for 8 weeks, look at the chronic adaptations, and then again do the acute. They'll do another strength training session or whatever it is.
After 8 weeks, of training, they'll do one session and see the difference from baseline to post.
So they have the chronic data and they have the acute data.
Right, and this whole field, people got to understand, it's. I think of the field of exercise science, and back in the 60s and 70s, it was all aerobic. It was a lot of looking at cardiovascular system, all that.
Yep.
Now, in the last couple of decades, it's really taken off where we're looking inside the cells, inside the muscle cell, at these different signaling pathways that cause different events and different effects, increases in muscle size. So this is something that's new. There's a lot of room for a lot of future directions and future research to take place, really, in this area.
Can I end with something? Just for the listeners, something is better than nothing. That's what we found.
Right. And again, I think it's interesting because I think of Dr. Brad Schoenfeld, like his research on volume and even the in 2021 the resistance training guidelines for muscle hypertrophy were published, and it was like, well. Now, again, we're talking here about an untrained individual. You can probably get by maybe with one set of each muscle group. But optimizing, for most people, I know like Schoenfeld, I think in his review, said you need about 10 sets per week minimum.
Yeah, yep.
So it's like there's a difference between growing some muscle versus optimizing the amount of growth you get.
Yep.
So
absolutely.
Yeah, do you have anything to say with those recommendations? 10 sets if you're wanting to optimize growth from muscle in a week, up to how many? 20 sets, 30 sets.
Yeah.
How much should
there's definitely an inverted U.
Right, because at some point you're going to get diminishing returns, right?
Yeah.
I
would say there's a lot of junk volume that happens in the gym that people don't need to be doing.
Yeah, I talked to someone at the gym several months ago, and I was like, "What are you doing today? What are you working out?" He's like, I'm doing 36 sets of chest today, and I was like, and that's where your junk volume. It's like, well, at some point you're getting diminishing returns, like it's not doing anything. So
chest would rip off.
I was like, why don't you just split that into 2 sessions? Maybe drop down to 30, maybe do 15 one day, rest 72 hours, do the other 15 later, or even, Schoenfeld, go down to 10. So yeah, there's a point, and it's hard to find this, and this is where there's so much debate in the literature as to what volume, when you're talking long-term training, this was an acute study, but what volume, where do you need to be at to optimize? Yeah.
We don't know, but I will, like I said, something is better than nothing. And, I think you can get away with 1 set versus 3 sets. As you progress, I think you're going to need more volume.
Right.
How much more? We don't know, but, I think more is better than 1, especially for us.
As you progress from untrained to more trained. Yes. Yeah. And that's why they call it progressive resistance exercise.
Yep.
You've got to progress some variable at some point.
If you want to get better, you have to.
Whether it's the load, or whether it's, it's either increasing the load, the intensity, the reps, the sets,
tempo, the rest. Yeah, sure.
Something has got to change, right? Because your body always wants to adapt to whatever stimulus whatever stimulus it's being given, and you don't want it to just adapt. You want to keep making progress.
Yeah.
So. Well, anything else? You want to add?
I think I summed it up. I think that's good. Yeah. I think we're good.
How can people contact you if they have questions about your research, training, bodybuilding, or nutrition?
Yeah. So guys, if you want to get a hold of me, if you have any questions, any concerns, or anything at all, if you want to talk to me, we can talk science. My Instagram handle is Jeremy R. Pearson, JEREMY RPEARSON. If you're interested in reading the paper, I'm more than welcome to send it to you. Or if you're interested in looking at my other papers or reading my other papers, you can Google my name and type in ResearchGate, and you'll find all the papers there. Or just go to ResearchGate and search my name there.
Google Scholar as well.
Google Scholar as well, yeah. Or just Google in general. If you type my name, it should pop up. Hopefully.
Okay. Yeah. Well, thanks for coming back on the show again today,
it was a blast.
Jeremy.
Thank you.
And if you have questions for me about the KU Exercise Science program, send an email to jtaylor@ku.edu or call 913-897-8516. Thanks again for watching another episode of Fitness Facts.